Namaste friends, welcome you all for the biology session. Let us continue with the chapter excretory products and their elimination. In the previous class we discussed about the structure of kidney and the internal structure by studying vertical section of the kidney. There we discussed different function, that is external cortex and inner medulla, which will be with different organs, or you can say different parts. Right now in today's class we can know about ultrafiltration and selective reabsorption tubular secretion. So these are the three steps which comes under urine formation. So urine formation, it generally takes place in the uriniferous tubules. Uriniferous tubules. So uriniferous tubules are also called as, we're all familiar with this, that is nephrons. Nephron is the structural and functional unit of kidney which helps in urine formation and elimination of nitrogenous waste. So urine formation includes three steps. One which is ultrafiltration, and the next step is the selective reabsorption, or you can say selective tubular reabsorption, and the third step is also, we can say that is also a selective tubular secretion. So here the main thing we have to keep it in mind, that what is the meaning of reabsorption and secretion. So ultrafiltration, which is also called as glomerular filtration, as it takes place in the glomerulus, hence it is called as glomerular filtration. So we'll take some very important points in this, and later we can solve questions related to this. So ultrafiltration, so whenever this step takes place, right, which takes place in the glomerulus, hence the name that glomerular ultrafiltration. This question we may expect, where exactly that the ultrafiltration takes place. So ultrafiltration takes place in glomerulus. Hence this glomerulus is called as the blood filter. Glomerulus na blood filter anta karitive. So what is the meaning of ultrafiltration? So if you consider that the structure of the Bowman's capsule, right, I think you're all familiar with this, that is receive the arteriole, that is mainly which will going to form a network of capillaries, right? So where this network of capillary once again join to form the arteriole. The arteriole which enters is called as afferent arteriole, enters the Bowman's capsule, and this is the Bowman's capsule. So if you consider this generally which is made up of two-layer structure, outer parietal and visceral layer. Outer parietal and inner visceral layer. So inner visceral layer is lined by simple squamous epithelium, and along with that, which has specialized cells called podocytes. We'll take these structures within a while. So podocytes are present seen in the Bowman's capsule, mainly in the inner visceral layer, right? So what is the role of the podocytes? Podocytes helps in ultrafiltration, right? Now here the pressure builds. Here pressure which builds due to the water present in the capillary. That is that pressure which will going to cause the filtration. So we'll take that. So what exactly the ultrafiltration? That is separation of small volume. So if you consider that in this step, separation of small volume of what is that, the solutes, small volume of solutes from large volume of what is that, the solute which will be there in the blood. So that separated, and some other components that gets what is it, the filter, right, due to filtration, right, through slit pores. These slit pores, which are also called as filtration slits. Slit pores are also called as filtration slits. These are formed by podocytes, are present within the podocytes. We'll take that structure, and here these are the epithelial cells, and there is foot like processes which are there, and that forms, these will be in the attached condition, right? So this is what exactly where we can say this slit pores can be present, through which what is it, the blood gets filtered into the Bowman's capsule. So this is the lumen of the Bowman's capsule. Hence, through that minute pores, the blood gets filtered. Hence, it is called as ultrafiltration. One is separation of small volume from the large volume, and by the through the minute pores, right? So in this case, generally in the ultrafiltration, so what happens? Let us take almost all the essential nutrients, both essential and non-essential components will be here. It is non-selective. It is a non-selective, what is it, the process where both essential and non-essential, solute, or you can say that the substances which will be gets filtered, hence it is called as non-selective. This is one question we can expect. And here, what is it, almost all the electrolytes, 70 to 80% of the electrolytes will be gets filtered, and almost all what is it, the glucose and amino acids will be reabsorbed. So whenever we're going to talk about this reabsorption, what it includes exactly? Reabsorption andre, keep it in mind that renal tubule, I already mentioned, any part of the renal tubule, PCT, DCT, endless loop, all in the renal tubule, the movement of solutes, right, from renal tubule, right, there will be what is it, the movement of solutes, into the blood capillaries, which will be peritubular capillaries, which is called as the peritubular capillaries, which are formed by efferent arteriole. So this structure, we can say this efferent arteriole, which they go and forms a peritubular capillaries, and that will be present surrounding to PCT and DCT, and same extend into the endless loop and forms vasa recta. So these are the two things you have to keep it in mind. If this apparent arteriole which branches and surrounds the PCT and DCT, that structure is called as peritubular capillaries. If it is surrounds the vasa recta, that is called as endless loop, that is called as vasa recta. So here these are the things, 70 to 80% of the electrolytes get filled, and all those things you have to keep it in mind. And where in the things we're going to say that the sodium and the potassium which will be absorbed by means of active transport, right? ಇಲ್ಲಿ ಎರಡು ಥರದ್ದು ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಆಗುತ್ತೆ, ಆಕ್ಟಿವ್ ಆ್ಯಂಡ್ ಪ್ಯಾಸಿವ್. ದಟ್ ಈಸ್ ಅ ವಾಟರ್, right? That is one kind of what is it, the passive transport, osmosis, and urea, little amount of urea, that is by means of simple diffusion, by means of simple diffusion which will be absorbed, simple diffusion which will be absorbed, reabsorbed. And here as I mentioned, what is the meaning of reabsorption andre renal tubule in the capillaries hogutte, that is solutes, movement of solutes. If from the capillaries, what is it, peritubular capillaries, or from the vasa recta, we can also say vasa recta, if solute, what is it, the movement into the, what is it, the lumen of the renal tubule, any part of the renal tubule, that is called as secretion. So that is why we'll say reabsorption and secretion. So here almost all the waste will be secreted into the renal tubule, and the essential will be absorbed from the renal tubule into the capillary. So this we can say, what is that, absorption. We can say absorption. Next, so next step is, what is that, generally selective reabsorption. Why it is called a selective reabsorption? This always takes place. Here two things you have to keep in mind, that is in the glomerular filtration both active and passive process will be, what is it, there by means of passive process, whereas in the, what is it, the selective reabsorption. So this selective reabsorption, right? So in this case mainly, which occurs by means of both active and, what is it, the passive. Here also we can say by means of both active and passive process, right? And the absorption which will going to takes place here in the PCT, mainly majority of the reabsorption takes place. This is one question we can expect. Majority of reabsorption, right, takes place in the, right, it is a seat of reabsorption, that is PCT. What is that? Here Na plus Cl minus glucose, and then what is it, the amino acids urea which will be absorbed. So here I will use a word that is absorption, right? ಇಲ್ಲಿ ಯಾಕೆ reabsorption ಅನ್ನೋ ಪದ ಯೂಸ್ ಮಾಡ್ತೀವಿ ಅಂದರೆ, ಫಸ್ಟ್ ಡೈಜೆಸ್ಟಿವ್ ಟ್ರ್ಯಾಕ್ಟಲ್ಲಿ this will be, what is it, absorbed, and once again that will be absorbed back into the blood capillaries. ಇಲ್ಲೂ ಕೂಡ ಬ್ಲಡ್ ಕ್ಯಾಪಿಲರಿಗೆ ಅಬ್ಸರ್ವ್ ಮಾಡ್ಕೊಂಡಿರುತ್ತೆ ಡೈಜೆಸ್ಟಿವ್ ಟ್ರ್ಯಾಕ್ಟಿಂದ ಕ್ಯಾಪಿಲರೀಸ್ಗೆ, ಅದನ್ನು ಅಬ್ಸಾರ್ಪ್ಷನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಮತ್ತು ಇನ್ನೊಮ್ಮೆ ಇಲ್ಲಿ, what is it, in the, what is it, from the lumen of the renal tubule into the capillaries, right? That is the mainly formed by the efferent arteriole. So that is called as reabsorption. And secretion which will be takes place like, what is it, the creatine and creatinine, we can say finally this will be converted into creatinine, some drugs and H plus ion, right, which will be secreted. Here I can use a word that is secretion. So next, PCT, and in DCT we can say that, here also we can say that is the, how exactly the absorption secretion takes place. Na plus Cl minus and K plus Mg plus H CO3, that is magnesium and carbonate ion, right, which will be reabsorbed. And here very important thing you have to keep in mind, that and little amount of ammonia along with the H plus ion and K plus ion and ammonium ion which will be, what is it, secreted, means which will be eliminated into the from the capillaries into the renal tubule. So these processes are very important, and in this the mainly here it will takes place. And in the Henle's loop, what is that, this which has a two limbs, we're all familiar with that, descending limb which permits the water whereas the ascending limb, right, ascending limb which is not permeable to water, descending limb is permeable to water but not permeable to electrolytes, not permeable to electrolytes, and the ascending limb is not permeable to water but permeable to electrolytes. So these are the things here you have to keep it in mind. And finally, in the collecting duct, what happens? So collecting duct, what is that? The sodium chloride, that is NaCl mainly, which will be reabsorbed along with little amount of urea to maintain the medullary, to maintain the the osmolarity in the medullary interstitium. Urea and most of the water along with that also, which will be reabsorbed. And here one thing you have to keep it in mind, that the collecting duct, right, which both collecting duct and DCT are under the control of hormones, that is aldosterone and ADH, right. Aldosterone helps in the absorption of sodium ion, that is electrolytes. ADH helps in the absorption of water. These acts on both DCT and what is the collecting duct. That is why we will say that the secretion, right, always takes place by means of active transport. So in the third step, that is the selective secretion. So what is that? In the selective secretion, you should keep it in mind, this is always the active process. So whenever the secretion takes place, so this here the can say always takes place in PCT and DCT, right. So mainly here, which includes almost all PCT, DCT, endless loop, collecting duct, but here only PCT and DCT are what is it, we're going to show the secretion. So secretion means, as we all already know, that will going to what is it, the tubular secretion, that is also selective. Only ultrafiltration is non-selective, keep it in mind. These two, what is it, tubular reabsorption and what is it, that is also selective, and secretion is also, tubular secretion is also selective. And here what is that, generally here you should keep in mind always by means of active transport. These are the questions we're going to expect. Active transport. And PCT and DCT, where there will be glandular cells, because of the presence of glandular cells, idu secrete ಮಾಡುತ್ತೆ, waste, excess waste will be secreted. And here in PCT, what happens, mainly PCT, which will going to takes the substances from peritubular capillaries, it takes, that is why the word we're going to use, what is it, secretion from the capillary into the lumen. It takes what is it, the almost all H plus, and then what is it, the NH3, that is ammonia, right, and ammonia which will be eliminated, and creatine, and sometime it will be converted into creatinine, and along with that little the amount of uric acid, right, and drugs which will be eliminated. And here the important thing we should keep it in mind, that all these will be, where is that, secreted right into the DCT. Next, DCT. In the DCT, what is exactly the secretion? Here you can say that H plus and K plus. This is the to helps in maintenance of ionic balance, and along with that helps in maintaining ionic balance, that is H plus and K plus ion. And whereas here acid base balance also we can say, and along with that the ammonium ion will be eliminated here, little amount of ammonia. So here very important thing you should keep it in mind, that only 15 to 20% of blood which will be gets filtered through nephron. Blood, what is it, the gets filtered, and that when blood gets filtered through the blood filter, that forms what is it, the glomerular filtrate. So this glomerular filtrate will be with only 15 to 20% of what is it, the waste material. 15 to 20% of blood plasma, in that 15 to 20% of waste will be gets filtered. So remaining what is it, the 75 to 80%, we can say, sorry, 80 to 85%, 80 to 85%, that is what which will be eliminated by this last step, that is called as secretion. If not, eliminate madak agala. So this is very, very important. So here just you keep it in mind, this is the, the structure of the nephron, right? And here it is less coiled, and here will be the collecting ducts, right? So I already mentioned that here endless loop, that is the descending limb. This descending limb is permeable to water. So just we can show like this, water, and the ascending limb is permeable to electrolytes, mainly Na plus Cl minus, which helps in maintaining osmolarity. And here keep it in mind that this descending limb is called as the concentrating limb, because water which will be moved out and the glomerular filtrate, here the filtrate which will be there, that is called as glomerular filtrate. So that gets concentrated. So this is called as concentrating the limb, or you can say that the segment, concentrating segment, right? And this one is when electrolytes will move out, that is diluting segment, right? So these are the two things you have to keep it in mind that. And when the concentrating segment and the dilute segment, if you consider, these are connected by the blood capillaries like this, it will be branched. This is what the vasa recta. So this vasa recta, and along with the limb of the the endless loop, together forms the special association, or you can say that the arrangement, right? And that which will be involved in this system, which is called as countercurrent system. So this is what you can say vasa recta, that is vasa recta and endless loop. So this is the descending limb of the endless loop, and this is the ascending limb of the endless loop, and this is the ascending limb of the endless loop, and this is the descending limb of the vasa recta. So here we can say that the ascending limb of the vasa recta in associate with the descending limb of the endless loop. So that is one, right? In the opposite direction the blood used to move. So this is what the counter, that is in the endless loop, descending limb downward to the medullary region, and the ascending limb from medulla to the cortex. Here also we can say that in the opposite direction. And if you consider, this is the vasa recta, and that is also which will going to show movement. This is the descending limb downward to the medulla, right, and the ascending limb outside the medulla, that is towards the cortex. So these are the questions. Here only water will be going to move, that water will move into the vasa recta, and here electrolytes will going to move from the interstitium, and then interstitium into the medulla, and that NaCl which will going to bring back, right, to the interstitium by this vasa recta. Hence vasa recta and endless loop together forms a system which helps in maintaining the osmolarity, or I can say osmotic concentration, right? That is what we can say countercurrent system. Countercurrent system, maintaining concentration gradient, and because of which the urine will be eliminated in the form of the concentrated or hypertonic urine. The countercurrent mechanism finally helps in the more reabsorption of water from the urine, that is the glomerular filtrate, and finally if water is more absorbed, only electrolytes, that is excess electrolytes and waste will be there, and that will be eliminated. So hence urine is concentrated urine. Finally the concentrated urine will be eliminated. Let us take some questions related to kidney structure and then we can go to this ultrafiltration. So main function of the kidney, as we all know that generally the kidney plays a major role in, so just now we discussed, generally in kidney they can also mention that the nephrons, so associated with, mainly they consist of nephrons. So synthesis of urea that takes place by the liver, whereas ultrafiltration, just now we discussed in selective reabsorption, both 2 and 3, what is that, is the function of kidney. Next, endless loop is found in. So if you're going to take that structure above that, here we can see that the Bowman's capsule in the part and here the little part, and endless loop will be there in the right. So if this structure, if you're going to say this is the cortex region and this is the cortex region and here it is medullary region, right? And if the length of the medulla, that is the length of the endless loop is more, right? If the length of the endless loop is more, right, then we can say more, what is it, the water reabsorption takes place. So if more water reabsorb, that the more concentrated urine, high concentrated urine can be eliminated. So that this type of will be there in the animals which lives in, what is it, the desert and where the xerophatic condition. Okay, next here, and this is the thing. So what is the answer here? This is the endless loop and this is the descending limb and this is the ascending limb. Next, so here this inner lining is lined by podocytes and podocytes with the, what is it, the filtration pores called as slit pores anta ಕರಿತೀವಿ ಅಥವಾ filtration pores. So this is thing you have to keep it in mind. Next, endless loop is found in, what is that, the not in lungs, not in heart, that is in kidney. Next, absent in liver. Next, 27, towards the center of the inner concave surface of the kidney. If you consider this is the kidney, towards the center, this part, inner concave of the kidney, there is a small depression, or we can say that the notch, right? I mentioned depression or notch, which is called as hilum, right? Through which the structures which will going to move into the kidney and some structures will going to leave the kidney. So that is, you can say, hilum, right? So what is that renal pelvis? We know that generally that is a funnel shaped structure which will be formed, that is the renal pelvis. And calyx, that is the medullary pyramids which will going to, what is it, the join to form a, what is it, the pyramids opens into the calyx, right? So this is what the thing in case of calyx. There are two types, major calyx and minor calyx. Columns of Bertini. So here you can say that the columns of Bertini, can say the place where, which will be present in between the, what is it, the renal pyramids, extension of the cortex is called as columns of Bertini, right? Next, length, width and thickness of the adult human kidney, that we discussed, right, in the previous class. And also there I mentioned we may get this question like this, the length and size of the kidney. So whenever we're going to take mainly result in this, so 10 to 12 centimeter in length, and then what is it, 5 to 7 meter, what is it, the in thickness, and 2 to 3 centimeter, 5 to 7 centimeter in diameter, sorry, that is 2 to 3 centimeter thickness. So that is the 10 to 12, you can say here 12 to 16 wrong, 12 to 16 to 12, and diameter is not 2 to 3, that is wrong. So here the answer is the first one, right? So length which will be vary. Next question we can take that the approximate number of nephrons per human kidney. So whenever we want to say that the approximate the nephron, there will be a 10 lakh the nephrons will be there in each kidney. That is 10 lakh, we can say that is 1 million. 1 million is equal to 10 lakh. So that is answer is third one, that is 1 million. Next, Bowman's capsule and glomerulus form. So as I mentioned that both Bowman's capsule and glomerulus, which forms a Malpighian body. So two parts of the nephron elidde nanu, Malpighian body, and one more we can say that is a renal tubule. Renal tubule andaga all those tubules of the nephron, which includes PCT, DCT, endless loop, collecting duct. It is two renal tubule. But this is the A Malpighian body, which includes these two parts, and renal tubule includes. So there are mainly two major parts in the nephron. One is a Malpighian body, adana Malpighian corpuscles antanu karitivi. Hage renal tubules, right, that we can say that the includes the tubular structure of the nephron. So that is the Malpighian body is the answer. And cortex is the functional structure where you can say that the outer zone, what is our outer region of the kidney, this is where this is the outer, what is it, the functional structure is called as cortex, and inner, this is the medulla. So in this medulla we can see that is these pyramids. So these pyramids are called as renal pyramids. So renal pyramids. So renal pyramids opens into the calyx, and calyx into the pelvis. So pelvis, which opens, passes through the hilum, and that forms a ureter. So this is the thing you have to keep it in mind. Next, brush border is a characteristic feature of. So as when we're going to see this structure in case of the Bowman's capsule, right? So I've already mentioned that, right, here the net negative pressure builds, right? So that is because of the glomerular hydrostatic pressure minus blood colloidal osmotic pressure plus tubular osmotic pressure, or you can say that the blood colloidal and tubular pressure. So this pressure, which will be going to builds whenever you can say that the you can say blood colloidal osmotic pressure, and the in this case this is the total net effective filtration pressure. Net effective filtration pressure. So which is also called as GFP, glomerular filtration pressure. ಹೇಗೆ ಬರ್ತದೋ ಗ್ಲೊಮರುಲಾರ್ ಹೈಡ್ರೋಸ್ಟಾಟಿಕ್ ಪ್ರೆಷರ್, ಇಲ್ಲಿ ಗ್ಲೊಮರುಲಸ್ ಇಲ್ಲಿ ಕೆಪಿಲರಿನಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಬ್ಲಡ್ಡಲ್ಲಿರೋ ವಾಟರ್ ಏನು ಪ್ರೆಷರ್ ಎಕ್ಸರ್ಟ್ ಮಾಡುತ್ತೆ ಅದು ಗ್ಲೊಮರುಲಾರ್ ಹೈಡ್ರೋಸ್ಟಾಟಿಕ್ ಪ್ರೆಷರ್, ದಟ್ ಈಸ್ 60 mmHg. And blood colloidal osmotic pressure, which will be there, the protein content which will be there, larger protein, they will going to the exert pressure against that water. So that is something around 30. And the tubular, or I can say capsular osmotic pressure, we can also say that the capsular osmotic pressure, so here this side, and these two acts as a back pressures. A 60 opposite of 50 total, that is this is the tubular osmotic pressure is 20. So finally 60 minus 50, so this is the back pressure. Back pressure is due to these two things. So that finally 10 mm of Hg in the filtration ಆಗ್ಲಿಕ್ಕೆ ಸಾಧ್ಯ. So that filtration ಆಗಬೇಕಾದ್ರೆ, right, whenever we're going to see this structure after getting filtration, as I have mentioned, ಇಲ್ಲಿ PCT ನಲ್ಲಿ ಆ ಫಿಲ್ಟ್ರೇಟ್ ನ ಗ್ಲೋಮರುಲಾರ್ ಫಿಲ್ಟ್ರೇಟ್ ಅಥವಾ ಪ್ರೈಮರಿ ಯೂರಿನ್ ಅಂತ ಕರಿತೀವಿ. ಗ್ಲೋಮರುಲಾರ್ ಫಿಲ್ಟ್ರೇಟ್ ಆರ್ ಪ್ರೈಮರಿ ಯೂರಿನ್. ಸೊ ಇಲ್ಲಿ this PCT, which shows the most of the major seat for reabsorption ಅಂತ ಹೇಳಿದ್ನಲ್ಲ, why? Because it is lined by cuboidal epithelium and the brush border appearance. More number of microvilli, densely arranged microvilli, which increases the absorptive surface area. Microvilli increases the absorptive surface area, and more mitochondria. These are the characteristic feature. More mitochondria and more microvilli Mitochondria provides energy, microvilli increases the absorptive surface area together, which will be there in the epithelial cells which lines the, what is that thing, which lines PCT, which lines, what is that, PCT. So that is why here I can mention the PCT is lined by, what is it, the cuboidal epithelium. So cuboidal epithelium. So what is that? Cuboidal epithelium with microvilli and, what is that, the microvilli which gives brush border appearance. Brush border appearance. So what is that? Brush border characteristics. Neck of the nephron. No, that is neck of the nephron means that is a Bowman's capsule. No, collecting tubule. It is not consisting of that the more microvilli, no microvilli and brush border appearance. Proximal convoluted tubule where we can see, what is that, the brush border appearance. Next, glomerular capsule. So whenever we want to say this, the structure of nephron lining kuda important generally. So where the active and passive both agutte largely, only now we can see this brush border appearance. If more only water in the Henle's loop, only water and electrolytes permeability will be there. So there we can't say that generally the brush border epithelium is present. Only cuboidal epithelium, simple cuboidal epithelium, no brush border, no, what is it, brush border, and very, if present, very sparsely arranged, scatterly arranged, what is it, the microvilli, and very less mitochondria, very less mitochondria. So these are the things you have to keep in mind. Glomerular capsule and convoluted tubules always lie within. So where exactly these structures will be present seen and so in this structure, if you're going to take this one, generally in which part ನಾವು ಹೇಳಿದ್ವಲ್ಲ ಫಂಕ್ಷನಲ್ ಸ್ಟ್ರಕ್ಚರ್, ಕಾರ್ಟೆಕ್ಸ್ ರೀಜನಲ್ ಇದೆಯಾ ಅಥವಾ ಮಿಡಲರಿ ರೀಜನಲ್ ಇದೆಯಾ ಅಂತ ಕೇಳಿದ್ವಿ. ಸೊ ಇಯರ್ ಗ್ಲೊಮರುಲಾರ್ ಕ್ಯಾಪ್ಸೂಲ್ ಆ್ಯಂಡ್ ಕನ್ವಲ್ಯೂಟೆಡ್ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಆಲ್ಮೋಸ್ಟ್ ಆಲ್ here you can see this, the structure once again I'll show. This is what cortex region, what is it, there is a DCT, PCT, and then collecting ducts also can be seen, right, all these. So when we can say in the cortex region, what is that, mainly the glomerular capsule and the convoluted tubules always lie within the, what is it, the cortex region, that is a renal cortex. Renal cortex. Renal pelvis is a, what is it, the funnel, what is it, the shaped a cavity, right, that is which receives the major calyces. Major calyces. Renal medulla, that is the functional one where we can see endless loop and little part of the collecting duct, right. Next, the fluid leaving which part of the excretory system of mammal can be called as urine. As I mentioned that in the PCT it is called as primary urine, and finally which leaves from the collecting duct. So in the collecting duct it will be called as generally it is urine, right. So idanna ನಾವು pyramid structure ನೋಡಿದ್ರೆ there is the collecting duct that opens through the papilla into the minor calyx. So here the things you have to keep it in mind. So collecting duct ಒಳಗಡೆ renal pyramid ಹೋಗಿ ಜಾಯಿನ್ ಆಗುತ್ತಲ್ಲ papilla, this is a renal papilla. Renal papilla. So here generally in the collecting duct it is called as, what is it, the urine. Next, which structure of the nephron is single, right. So which structure in nephron is single? So generally that vasa recta is a network of, and the convoluted tubules, you can see that the convoluted tubules, proximal convoluted tubule, DCT, distal convoluted tubule, not single pair are there. And the collecting duct, so whenever we're going to see this, the many collecting ducts that opens, right, there are many collecting ducts which receives the opening of the, what is it, the connecting tube of the many nephrons. So that is also we can't select. And Bowman's capsule, only one. So if you're going to count the number of Bowman's capsule so that you can what is the count in number of what is the nephrons. So number of nephrons can be easily counted by counting Bowman's capsule. So that is what this single, and vasa recta is a branch of capillary, what is it, the, or you can say capillary network. That is which also has the descending limb of the vasa recta and ascending limb of the vasa recta. Hence even we can't select this. So answer for that is Bowman's capsule. Next. So the projection of renal pelvis are called as, what is that? So whenever we're going to say about a pelvis, renal pelvis, which is also called as the pelvis, the projection of renal pelvis is called as what? What is the meaning of that? The renal pelvis. So there from this projections which receives, which is a projection of renal pelvis. This is a pelvis which forms a ureter. Right, through the hilum it passes out, and here these projections are called as, which receives the smaller calyces, that is a minor calyces, and this is called as major calyces. So projection of the pelvis, renal pelvis, are called as major calyces, and major calyces forms, and where you can see these things, medullary pyramids which will be attached. So this is what generally you can see the structure, and answer for that is the projections of the renal pelvis are called as calyces, major calyces and then minor calyces. So answer for this, and hilum is a notch in the concave region, center of the kidney, where there is a concave region. Renal columns, right, which will be there in between the medullary pyramids. Renal columns in between the medullary pyramids. So this we can say renal columns and medullary pyramids, and these are the medullary pyramids. Next, which of the following structure facilitates easy passage of small molecules to the lumen of the Bowman's capsule? Only small molecule. So as we already discussed that there is a membrane. So let me give that membrane name is filtration membrane. When the blood gets filtered from the right, this is the glomerulus, right? So glomerulus in the blood ಏನು ಫಿಲ್ಟರ್ ಆಗುತ್ತಲ್ಲ, ಇಲ್ಲಿ ಪ್ರೆಷರ್ ಆಗುತ್ತೆ, ನೆಟ್ ನೆಗೆಟಿವ್ ಪ್ರೆಷರ್. ಸೊ ನೆಟ್ ಎಫೆಕ್ಟಿವ್ ಪ್ರೆಷರ್ ಹೇಳಿದ್ವಿ ನಾವು 10 mmHg. So that is what glomerular filtration pressure, which is also called as glomerular, the blood capillary pressure. Glomerular blood capillary pressure ಅಂತಲೂ ಕರಿತೀವಿ. ಈ ಪ್ರೆಷರ್ ಮುಖಾಂತರ, ದಟ್ ಈಸ್ ಫಸ್ಟ್ ಗ್ಲೋಮರುಲಸ್, ಇದು capillary lined by what ಅಂತ ಕೇಳಿದ್ರೆ, so generally this capillary is lined by endothelium. That is endothelium of the capillary, filtrate passes, and the next here in between there is a basement membrane. And this is the the basement membrane, right? Both will going to have their own basement membrane, but together we're going to consider basement membrane for the endothelium, basement membrane for the squamous epithelium. These squamous epithelium with a specialized podocytes, specialized cells called as podocytes. Like this, the arrangement, intricate, the arrangement, delicate, ತುಂಬ ಕಾಂಪ್ಲೆಕ್ಸ್ ಒಂದಕ್ಕೊಂದು ಕನೆಕ್ಟ್ ಆಗಿರ್ತವೆ. ಈ ಪೋರ್ಸ್ ಏನಿದೆಯಲ್ಲ, ಇದರಿಂದ ನಾವು ಫಿಲ್ಟ್ರೇಷನ್ ಪೋರ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಇದರಿಂದ ಬ್ಲಡ್ ಪಾಸ್ ಆಗಲಿಕ್ಕೆ, ಬ್ಲಡ್ ಪ್ಲಾಸ್ಮಾ, ಇಲ್ಲಿ ಓನ್ಲಿ ಸ್ಮಾಲರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಆ್ಯಂಡ್ ನೆಗೆಟಿವ್ ದಿ ಅಯಾನ್ಸ್, ರೈಟ್, ದಿ ಕ್ಲೋರೈಡ್ ಅಯಾನ್ಸ್ ವಿಚ್ ಇಸ್ ಅ ನೆಗೆಟಿವ್, ರೈಟ್, which will be negative won't enter generally. That is only a small, as it is a smaller, right, that is why it will going to get the passes through. So this is very important. Intricate manner ಎಷ್ಟು ಮಟ್ಟಿಗೆ ಕನೆಕ್ಟ್ ಆಗಿರುತ್ತೆ ಅಂದರೆ it allows only smaller molecules except those platelets, and it won't allow platelets and larger proteins ಯಾವುದೇ ಕಾರಣಕ್ಕೂ allow ಆಗಲ್ಲ ಇಲ್ಲಿಂದ. ಅದಕ್ಕೆ ಎಂಡೋಥೀಲಿಯಂ, next layer is a basement membrane, and next layer is the squamous epithelium of the Bowman's capsule. ಇಷ್ಟು ಸೇರಿಸಿ, ಸೊ ಫಿಲ್ಟ್ರೇಟ್ ಇಷ್ಟು ಲೇಯರ್ ಮುಖಾಂತರ ಪಾಸ್ ಆಗುತ್ತೆ, ಅದನ್ನು ಫಿಲ್ಟ್ರೇಷನ್ ಮೆಂಬ್ರೇನ್ ಅಂತ ಕರಿತೀವಿ. ದಟ್ ಲೇಯರ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಫಿಲ್ಟ್ರೇಷನ್ ಮೆಂಬ್ರೇನ್. ಸ್ಕ್ವಾಮಸ್ ಎಪಿಥೀಲಿಯಮ್ ಆಫ್ ದಿ Bowman's capsule with podocytes. Epithelium of Bowman's capsule with podocytes. So answer for this, generally whenever the structure which will be going to helps in that, so here filtration membrane, because of this filtration pressure, right, blood plasma which will going to move here, that blood plasma with all those essential and non-essential, that is why here it is called as primary urine anta kariyalla, only waste urine composition node. Okay, so next in this, what is it, we can see the questions like, so we'll take next question, what is true about excretion. So in this case, the questions we will going to see, generally whenever the elimination will be there, ಏನಾಗುತ್ತೆ, ನಾವು ಹೇಳ್ಬೇಕಾದ್ರೆ ವೇಸ್ಟ್ ಮೆಟೀರಿಯಲ್ನ ಎಲಿಮಿನೇಟ್ ಮಾಡುತ್ತೆ, ಆಲ್ಮೋಸ್ಟ್ ಆಲ್ ಎಸೆನ್ಷಿಯಲ್ನ ರೀಅಬ್ಸರ್ವ್ ಮಾಡ್ಕೊಬಿಡುತ್ತೆ. ಎಸೆನ್ಷಿಯಲ್ ಅಂದರೆ ಯಾವುದು, ಗ್ಲೂಕೋಸ್ ಆ್ಯಂಡ್ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಆಲ್ಮೋಸ್ಟ್ ಆಲ್ ಅಬ್ಸರ್ವ್ ಮಾಡ್ಕೊಂಡ್ಬಿಡುತ್ತೆ, ರೈಟ್. ದಟ್ ಈಸ್ ಯೂರಿಯಾ ವಿಲ್ ಬಿ eliminated, but in that part of urea which will be reabsorbed back, mainly to maintain the osmolarity in the medullary interstitium. Osmolarity in the medullary interstitium. So 90% of the water and sodium ions of glomerular filtrate are absorbed. So 90%, that is 70 to 80% of the electrolytes which will be absorbed. 70 to 80% of the electrolytes which will be absorbed. So not 90%. So that is, we can't select that answer. And next, glucose is reabsorbed in distal convoluted tubule. No, as we discussed in the ultrafiltration, after the ultrafiltration, tubular reabsorption that takes place in PCT, urea is not at all reabsorbed. No, it is reabsorbed, as I mentioned, to maintain the osmolarity of the medullary interstitium. Medullary interstitium, right? So there it will be reabsorbed. 99% of the water in the glomerular filtrate is reabsorbed. So this is correct, right? And almost all glucose and amino acids also reabsorbed. So this is a correct one. Next, the principal nitrogenous excretory compound in human is synthesized in, where exactly the waste will be synthesized. So here we can say that generally the liver, right? So but that will be eliminated through kidney. See, synthesis of this nitrogenous waste with the help of ornithine cycle, mainly that is the urea, takes place in the liver, right? Liver which synthesize the urea but eliminated by kidney. ಅದಕ್ಕೆ primary principal organ, right? Primary principal organ for excretion ಅಂತ ಹೇಳಿದ್ರೆ generally we can say that is ಪ್ರೈಮರಿ ಪ್ರಿನ್ಸಿಪಲ್ ಆರ್ಗನ್ ಅಂದರೆ ಕಿಡ್ನಿ ಅಂತ ಹೇಳ್ತೀವಿ, ರೈಟ್. ಹಾಗೆ ಪ್ರೈಮರಿ ಪ್ರಿನ್ಸಿಪಲ್ ಆರ್ಗನ್ ವಿಚ್, ರೈಟ್, ಸಿಂಥಸೈಸಸ್ ದಿ ನೈಟ್ರೋಜನಸ್ ವೇಸ್ಟ್ ಅಂದರೆ, ಪ್ರೈಮರಿ ಪ್ರಿನ್ಸಿಪಲ್ ಆರ್ಗನ್ ವಿಚ್ ಸಿಂಥಸೈಸಸ್ ದಿ ನೈಟ್ರೋಜನಸ್ ವೇಸ್ಟ್ ಅಂದರೆ ಲಿವರ್. ಲಿಟಲ್ ಅಮೌಂಟ್ ವಿಲ್ ಬಿ ದೇರ್ ದಿ ಸೆಕೆಂಡರಿ, what is the principal organ which synthesizes mainly the nitrogenous waste ಅಂದರೆ little, only little which will be there from the kidney. So that is why the answer for the kidney won't synthesize, wrong. Eliminated through liver, wrong. Kidney as well as, this is also wrong, as well as eliminated by kidneys. Production valle, eliminated valle, no. Liver and also eliminated by the same through the kidneys, that is correct. Liver also eliminated same through the kidneys. So here we can say that when the waste which will be produced, that will be eliminated, right, eliminated by the same through, that is we can say bile juice, right? So not through kidney, bile juice. So that is wrong. Only bile pigments will be eliminated. Bile pigments through bile juice. Okay, so typing mistake, bile juice. They are wrong. Liver, but eliminated mostly through kidney. So answer for that is, what is that? Fourth one, right? So principal nitrogenous waste produced in the liver, eliminated by kidney. Next, which of the following is not, what is it, the integral part of the nephron? Under a separate part ಆಗಿರುತ್ತೆ. So which is not an integral part, glomerulus, what is it, the Henle's loop, right, and then distal convoluted tubule, loop of Henle. See, it is not belongs to confined to one nephron. So as I all mentioned that Bowman's capsule bandide, but E Bowman's capsule, all those PCT, DCT, Henle's loop, and even though collecting duct, right, it is the associated with, right, if this is a thing, it is associated with, right, complete nephron. But what is that glomerular? What is it? The glomerulus? That is a network of capillary, the circulatory system sambandhapattidu, which enters the capsule. So that is what you have to keep it in mind. So renal artery enters the kidney. When it enters the kidney, that divides into arterioles. Those arterioles enters the Bowman's capsule as afferent arteriole, and leaves the kidney as efferent arteriole. Afferent arteriole ಏನಿದೆ ಅದರ diameter ಜಾಸ್ತಿ ಇರುತ್ತೆ. Efferent arteriole diameter will be very less. So when the content which move, blood along with plasma which will move, ಇಲ್ಲಿ ಪ್ರೆಷರ್ ಜಾಸ್ತಿ ಬರ್ತಿದ್ದಾಗ, ರೈಟ್, ಇಲ್ಲಿ ವಾಲ್ಯೂಮ್ ಜಾಸ್ತಿ ಬರ್ತಿದ್ದಾಗ, ದಟ್ ಈಸ್ ಇನ್ ದಿ ಅಫರೆಂಟ್ ಆರ್ಟರಿಯೋಲ್ ಏನಾಗುತ್ತೆ, pressure here when the narrow the lumen is there, narrow lumen, that will going to build up pressure, right. What are all those water molecules will going to build up pressure, protein will going to, that is what proteins present in the plasma will going to build up pressure, and against that in the capsule is also there will be, what is it, the content, right. It is not like it will going to completely empty and then it will be going to fill. No, there will be a continuous movement of solutes, and these waste will be there. And here also some that the colloidal, what is it, the solutes will going to exert a pressure. So that is what totally will going to form net effective filtration pressure, which is also called as glomerular filtration pressure. ಇದು glomerulus, ಇದು ಇಂಟಿಗ್ರಲ್ ಪಾರ್ಟ್ ಅಲ್ಲ. ನೆಕ್ಸ್ಟ್, ನಾರ್ಮಲ್ ರೇಂಜ್ ಆಫ್ ಯೂರಿನ್ 100 ml, ನಾನು ಹೇಳಿದೆ ಎಮ್ ಜಿನಲ್ಲಿ ಹೇಳ್ತೀವಿ, ಎಲಿಮಿನೇಟ್ ಮಾಡಿದಾಗ ಗ್ರಾಮಲ್ಲಿ ಹೇಳ್ತೀವಿ. ಸೊ ದಟ್ ಈಸ್ ಎಲಿಮಿನೇಷನ್ ನಾವು ಗ್ರಾಮಲ್ಲಿ ಹೇಳ್ತೀವಿ, ಆ್ಯಂಡ್ ಎಮ್ ಜಿ ಅಂತ ಹೇಳಿದಾಗ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, ದಟ್ ಈಸ್ ಐ ಮೆನ್ಷನ್ ದಟ್ ಸಮಥಿಂಗ್ 15 to 30, ವಿ ಕ್ಯಾನ್ ಸೇ 17 to 20 mg, what is that, mg, milligram per 100 ml will be there, and that will be eliminated. Next, in this case we can expect, that is the question here, the composition of, what is it, the urine. Whenever we are going to study about the urine content which will be there, that is mainly, what is it, the content of the urine, you have to keep it in mind that urea will be there, and then more majority of water will be there, and that the urochrome is the pigment produced during the degradation of, what is it, the RBC only, right, and that will be from the liver, that will move back to the intestine. From the intestine it will be reabsorbed into the, what is it, the kidney, where we discuss intestine into the kidney, renal portal system. So renal portal system, and that urochrome, which the pigment which imparts a yellowish color to the urine, So characteristic color is because of the urochrome, presence of urochrome, and that will be eliminated, what is the waste through the urine. And next question we can take, uriniferous tubules occurs in, uriniferous tubules are also called as nephrons, right. So they will be present seen in kidney, right. In stomach we won't see, ovary, no. What is that? In testis we can see seminiferous tubules. These seminiferous tubules which helps in the production of sperm, spermatogenesis. Glomerular hydrostatic pressure, right. So glomerular hydrostatic pressure, right. So whenever we are going to say that, generally this pressure which will be exerted mainly, as I mentioned, because of the water present in the glomerular capillary. So here we can make it this none of the above, right. So renal tube, no, there will be what is it, the, we can say tubular, or you can say tubular pressure, right, and here glomerular capillaries, right, in which the glomerular hydrostatic pressure will be seen. So that is what GHP, the glomerulus, that is by water present in the plasma, right. Next, most probable cause of heart attack. So this is actually related to the chapter that is body fluid and circulation, and we can also say that here, due to the accumulation of the waste, which may leads to or cause the death, or you can say that. So what is the answer for that? Systolic pressure, that is normal, this is 120. Motion, that is also no. High level of HDL, no. Arteriosclerosis, that is the blockage, right. So blockage of what is it, arteries, right, becomes because of the accumulation of the calcium, right, that becomes gets thickening, right. The inner wall layer gets thickened, and that can't be able to show what is the proper constriction and relaxation. So because of that, the less amount of oxygen will be moved, right. So less availability of oxygen to the heart, that leads to heart attack, right. Generally, this is the thing. Which blood vessel takes blood away from the glomerulus? So name the blood vessel which takes blood away from the blood vessel. So as we all know that I already mentioned, this is the afferent arteriole which enters the Bowman's capsule and forms a network, right, that is efferent arteriole. So which blood vessel takes blood away from the glomerulus? That is efferent arteriole. Into the glomerulus, that is afferent arteriole. Out of that, efferent arteriole. So answer for that is 3. Answer for that is efferent arteriole, right. So keep in mind, the diameter is more here, more than the efferent arteriole. Here lesser diameter than the afferent arteriole. That is why the pressure builds. One reason, that is one reason, along with glomerular hydrostatic pressure, that is what, minus blood colloidal osmotic pressure and capsular osmotic pressure or tubular osmotic pressure. So this is what we can say, that the glomerular filtrate pressure. So glomerular filtrate pressure is this because of 10 mm. Collecting ducts extends from. So from where to where these collecting ducts are extended. See this region, once again you can relax, that is a connecting which opens, and there further that which is extended from cortex to the medulla. Cortex to the inner medulla, right, whereas a Henle's loop present seen in the medulla, right. So this is the PCT and this is DCT, right. Here one more thing we can say, PCT is highly coiled, more brush border appearance than, and then this is slightly coiled. So these are the things you have to keep in mind. So from cortex to inner medulla. Sir, what is that inner medulla? This region, if this is a cortex and this is the medulla, in that medulla we can divide into, that is the outer medulla and inner medulla. So from outer to inner, what is it? The osmolarity increases. So here outside that will be 13, what is it? The osmol per liter will be there, 300 osmol per liter will be there, and here it is going to increase 400 to 600 and then 800, totally inside 1200 milliosmol per liter will be there. So this is what osmolarity, because of the NaCl and urea which will be absorbed here, and that will be moved back to the interstitium from the collecting duct, and here water will be removed, and here NaCl which will be absorbed, that will be moved out, and that helps to maintain of, that is what a counter current mechanism, right. Next, the amount of glomerular filtrate formed. So generally, whenever we can say that we can also mention that in the 250 ml per minute, or you can say that 180 liter per day, right? So these are the things you have to keep it in mind. Next, so that is 180 ml. Correct sequence of urine formation. So first 3 steps are there: ultrafiltration, 3 steps, ultrafiltration, that is also called as glomerular filtration, reabsorption, and then secretion. Filtration, reabsorption, and secretion. So first one, filtration, reabsorption, secretion. Reabsorption, this is second step, wrong. Filtration, micturition. Micturition means urination. Urination, right? So that is wrong. Filtration, reabsorption, diffusion. No. So answer for that is next. Which of the four parts mentioned below does not constitute part of single uriniferous tubule? That is what? As it receives uriniferous tubules means many nephrons. Many nephrons received by collecting duct. So collecting duct is the formed by receiving many connecting ducts of the nephron. So that is what the answer, collecting duct. Single uriniferous tubule, right, does not constitute a part of single. The collecting duct is not belongs to a single nephron, it belongs to many nephrons. Next, cells named podocytes occur in. So I already mentioned that the podocytes, they are present in the Bowman's capsule, mainly in the visceral layer of the Bowman's capsule. Visceral layer of the Bowman's capsule. So that forms a sac-like structure, right here. This is what, and with those 2 layers which will be open, that is a blind end. Blind sac. Here is an opening and here there is no opening, that is covered, right? So that is Bowman's capsule, right? Neck of the nephron means Bowman's capsule. Wall of the capillaries, that is the mainly endothelium and glomerulus of kidney, that is formed by the afferent artery. Network of capillaries formed by afferent arteriole. So next, filtration slits are formed by. So already I mentioned that the structures which will be there like this, foot-like projections, are called as pedicels. These are called as pedicels, and in between the pedicel there will be a minute pores, and these pores, they are going to open into the lumen of the capsule. That is what generally filtration slits or slit pores. Filtration slit, which are also called as slit pores, present seen in the Bowman's capsule, mainly the inner epithelium of Bowman's capsule. So answer for that is endothelial lining of the glomerular capillary, no, basement membrane in between these two, right? In this 50th answer is this one. So let us continue next questions in the next part.